Thank you for this very kind introduction and raising expectations. So I hope that I will kind of manage to hold this expectation and kind of convey the message about my research and make it interesting for everyone in the room. So as what I would like to talk about today is my pilot study funded by BRACE. But I also would like to kind of put it in a broader context, exactly what I just said right now. So my background, which is computational neuroscience and looking at images and different models of brain organization and brain network, and how that kind of relates to this pilot study funded by BRACE. And also, because I'm health data scientist, and in health data, integrating information from different health-related data with the neuroimaging in order to better understand dementia. And my research, in a way, in dementia and neuroimaging revolves around these two questions, which are in a way two sides of the same coin. One is how we can, what we can basically learn from patterns of atrophy in or cortical changes in dementia neurodegeneration, which is not random but organized into some different structures, and using networks to understand how this organization actually emerges from neurodegeneration, but then also to understand how we can look at the individual differences in healthy aging in terms of changes in the brain and in the cortex, to understand why some people get Alzheimer's disease or any other type of dementia, and others stay healthy or without any brain changes or smaller brain changes, which basically do not or impair their cognition and function. So because I did say I'm going to talk about my research is about building models, neuroimaging models, and looking at the data set. So what we would like to achieve with this model, brain is extremely complex organ, as we already all know, and we somehow hope that we can that we will be able to build this type of model from or map of the brain, which is a London Underground map, and it has this is perfect map of the complex network of the underground, because it serves its purpose. It has detailed enough information for someone who go from A to B. There is not too many details, there are no landmarks and so on, but then also it is only representation and a map. So we basically would like to build a model that is similar to this model in terms of information contained in the model. So that's why we can build all the connection. There are all the connection. There are stops, lanes, and everything else that we need to just move through this transportational network. So in next few 45 minutes, I would like to just start. This is the outline of the talk, and I would like to start with some early neuroimaging studies to show how patterns of atrophy in Alzheimer's disease and frontotemporal dementia, which is a different type of dementia that I was also looking at and working with. And then how we basically used integrated approach, looking at imaging, genetics, histological pathology and clinical scores to understand patterns of atrophy and then relate to different type of this information and data. And then why do we use brain network models to explain these patterns of atrophy, what we can basically gain from using networks rather than looking just at sizes of the certain cortical areas or size or of changes. And then I will also talk about Brace-funded study and give some preliminary results. We have some data and already on medRxiv, some published, not peer-reviewed. That's something that I'm going to talk about. So this is a first early view on imaging, neuroimaging data in Alzheimer's disease, typical Alzheimer's disease and behavioral variant frontotemporal dementia. This study is from 2013. But we now understand much more about this disease. So what we see here is yellow areas in the typical Alzheimer's disease show atrophy or neurodegeneration in these regions. But in frontotemporal dementia, which is another type of dementia, atrophy patterns or atrophy changes in the cortical regions are located in frontal and temporal regions, what the name says. There are obviously some similarities because in both diseases we see that temporal lobe is affected in terms of what atrophy compared to control. So all of these data based on looking at the differences between age-matched healthy controls and patients with typical Alzheimer's disease or behavioral variant frontotemporal dementia. So in terms of this, these are early picture of the atrophy patterns in these diseases. But what is interesting in compare from Alzheimer's disease and frontotemporal dementia, we always see Alzheimer's disease that affects posterior brain region and temporal regions. But frontotemporal dementia, even then, like 10 or 15 years ago, was always seen like more heterogeneous, even in terms of atrophy. In this case, this is behavioral variant. And what was interesting about this type of atrophy pattern in behavioral variant that we can see from T1 MRI images here, which are basically available in everywhere in NHS clinics, and they are very routine clinical scans, and it's not difficult to get MRI scan compared to PET, for example, from what Liz even also mentioned. So that's why we were looking at the T1 scan rather than looking at some different one. They are really good because they provide information about brain structures, about morphology, and they have very good spatial resolution. This is a healthy subject on the left, and these are two patients with behavioral variant frontotemporal dementia. So what is interesting about these two patients is that, and why I choose to show them, is that their clinical scores, in this case MMSE and ACE, this Sutherland score, and two others are behavioral scores, are same, but what we see from their images that these dark regions in frontal lobe are different for these two patients. So this is obviously interesting research question. How does two patients with the same clinical scores have different patterns or different neurodegeneration in different regions from looking at their MRI scans. The question was how we can we just classify these patients based on their differences in different regions, actually patterns of atrophy? How we can, is there a way just to classify them to see, is there some of them showing similar atrophy in similar regions or different ones? So what we have done, we were looking at, we had 200 participants, and at the beginning of the study, we parcellated their frontal and temporal regions and then used to compare, looking for similarities and differences. And then we found that there are actually, that they cluster in 4 clusters. In each of these clusters, these are like portion of participants of a portion of patients in each of the clusters. And then when we compare clusters with healthy control, age-matched healthy control, we then realized that the first group is frontotemporal parietal, shows this kind of overall very, very broad and very, very broad atrophy here in yellow color. And then the second cluster is temporal dominant with temporal regions dominating. There are more atrophy in temporal region than in healthy subjects. And then frontal dominant is third one, and then sublobar is another cluster of participants. But what is also interesting about this clustering is that even though they show differences in this cortical surface, obviously, there are some core regions, in total around 15 of them, with there are no differences. So there is atrophy in all of in every single region, and almost 10 of these regions are actually deep brain regions. Insular cortex, anterior cingulate cortex, amygdala, and all these deep brain regions, like anatomical nuclei. And then we then mapped these regions onto known cognitive and behavioral network, and most of them are most of them are basically part of the so-called salience network, which is the behavioral network responsible for responding to salient into external task, in switching behavior from internal to external. So that means that even though there are different patterns around the cortical surface, they still show that as core regions which correspond to salience network. So if you remember from this core from the beginning of the talk, we then also classified or divided the scores into cognitive and behavioral domains. And then we found that, for example, temporal dominant subcluster or cluster kind of dominates this cognitive subscore, or it shows relationship between with cognitive scores of semantic memory, language, and fluency, letter fluency. Most of the time, temporal dominant subcluster, which we would expect to find temporal dominant, given the knowledge that most of the time language region and semantic memory regions are located in temporal lobe. But then at the same time, behavioral scores divided into their own domains. In this case, we found that frontal dominant cluster kind of dominated this part of the of the map. And that most of the time frontal dominant is associated with any of these core, from apathy to disinhibition, and then also composite final score, clinical score. So this is not now our only finding. There are other group who has done similar comparisons. So on the left side are all different types of frontotemporal degeneration, and then on the left is radar plot, so where you can see how different, so light yellow and red are region degenerated or atrophied, and then how they map onto their cognitive or behavioral scores. And even those four with here tauopathies, which are all certain type of tauopathies, but with different type of tauopathies, they also show different patterns of atrophy, and then also different maps with the behavioral score and different direction in each of the radar plot. And the last one was AD, like second from the bottom is Alzheimer's disease, which is mainly hippocampal atrophy, but there is no relationship, there is no kind of mapping to behavioral score, because they are all within the this small area under the radar. And this looks like that picture of the Alzheimer's disease is similar, stays similar, but there are no heterogeneous patterns of atrophy in Alzheimer's disease. But some studies from Hebbe's group in 2020, they also looked at Alzheimer's disease and compare with the healthy control, and then also found that these are all with the CDR score, similar CDR score. So this is not these differences are not because they're at the later stages or so. So what we see is that they also have something that is typical AD, something that I also show. Every atrophy is at the cortical surface, a different area, but then there is only the second one is limbic subtype, and then hippocampus pairing, which the hippocampus is paired, but there are some patches of atrophy around the hippocampus, and the one there is no atrophy at all. And then the bottom plot shows how hippocampus, only size of hippocampus between these subtypes, all of them has reduced low. So this is size of hippocampus in healthy control, and then in all other subtypes is reduced and smaller than healthy control, and except this is where hippocampus is paired when the levels are very similar. But then this is from T1 images. Okay, another. This is from T1 images, anatomical MRI scans. But then we see very similar patterns and very similar situation if we look like amyloid beta images and try to cluster these participants or these patients compared to their differences with the healthy control. And there are similar 3 subtypes, one which is called typical, and they look very similar, and then limbic and cortical predominant. So basically, if you look at the different modalities, we somehow see very similar patterns, but then we obviously have different information based on modalities we use, and what are the differences between different imaging modalities, because these patterns are organized, they are not random, but they are also similar and different. So in a way, we need to find a way to quantify differences and similarity within using different imaging modality or combine different imaging modality to understand better or either mechanism or how, what actually is underlying pathology, or just to relate these two. So it's usually amyloid starts to accumulate and how it then spreads, and then there is neurodegeneration. So that's also something that Liz showed you in her slide, in a way, how everything is shifted in time. And now how we actually relate this to aging and making what aging has to do with patterns of atrophy, and how we can relate aging with vulnerability different brain regions with patterns of atrophy. So I was looking for how I actually present this and how do I find like a young adults and then show very similar way, find very similar way, like young and then older, and what are the differences. But I just couldn't find something that is very illustrative. So I decided to present this. This is some very interesting research field currently, which is called brain age. And because total brain volume and brain shrinks as we get older, but that's not with equal rate with all people. And we know that some people obviously, there are differences, and this is just like total brain volume. So if everyone, this is X, Y is brain age or biological age given as a total volume, and this is age, chronological age given in years. So if everyone brain age is the same way, we will have all points here along this line. But what we see, if there is a distribution, there is a variability, and then some people appear older than their chronological age, and then some appear younger compared to their chronological age. So this also shows that brain age is in a different way, just like looking at the total brain volume. And then how now networks come into play. So we obviously, if we just look at size of the regions and only how they shrink, that is still a very traditional way of looking at the differences. We saw obviously that there is a pattern in a way how region shrinks, and there is also a pattern in a way how disease progresses. And in this, so the question is, and there is a level of organization in this pattern. And then the pattern, then the question is there anything, how, for example, this intrinsic brain organization and how different connections in the brain, underlying connection affect how progression looks like, size of the region, connection between them, and how, for example, in this case, if you look at frontal parietal or occipital lobe, we know that closer regions are more likely to be connected. But then we also know that region between different lobes are also connected. And then we can use MATS or graph theory or network sites to quantify these connections and quantify pattern and quantify how is everything organized. We can also look at under cortical surface and looking at the layer level and see how connections are between different cortical layers are organized and happening. So that is the obviously beauty of network science in terms of that is that we can look at properties of the brain as a network at different scales. For example, as a spatial scale, looking at a cellular level interaction between neurons or different types of the cell in the brain, or populations, or here looking at how the connections are happening at the regional level or at the whole brain level. And then there is another scale, which is a topological scale, or how connections are organized. Again, we can look from local connections between, for example, pair of regions, and how they form and quantify. We can also look at motifs or modules and look is there more connections between certain collection of regions or outside, and then look at some other more global properties of the network in how information flow in the network, or basically depending on the research question, you can look at the different properties of the network. And there is also another component of scale, which is temporal scale. We can look from EEG recordings, which are millisecond recordings, then the changes that happens in the brain in using fMRI from several seconds, then developmental and development on the more on the larger scale. And so basically all these scales, we can use network science to look at the brain and at all these different scales. So that is the beauty of network science. And something that we have done is to look at integrating like genetic data from database, well-known database, with and look maps of the genes in the brain, and then look at fMRI data and try to associate and look how mapping genes in the brain are associated with functional networks. So we decided to look at Alzheimer's disease genes, around 80 variants associated with Alzheimer, and also cholinergic pathway genes, around 30 variants associated with cholinergic pathways. And there are no overlap between cholinergic pathway genes and Alzheimer's disease. But what was surprising is that map, so that gene expression in brain region looks very similar for Alzheimer associated and for cholinergic pathways. We only see a few differences, for example, here and here. And there is also similarity in terms of lateralization. There are more both Alzheimer associated and cholinergic pathway genes associated with left than right hemisphere. And most of the region that we mapped out with gene expression with Alzheimer's disease are part of so-called default mode network, which is well involved in self-referential processing. And then it's also well-known hallmark or biomarker, decreased activity or decreased functional connectivities. The DMN network is biomarker of Alzheimer's disease. So we then correlated, looked for gene expression in this region and how is there a trend. So there is a trend with the gene expression in the region and importance of this region in DMN network, which we quantify by eigenvector centrality, or the measure that tells you how important node is in the network and how many connections it has. Go back to T1 images now. Everything I told you so far is from T1 images, and also functional images is looking at the volumes, brain volumes, and extracting volumetric data. But T1 images are really good, with good resolution, and have like plenty of other information about the brain. In most intuitive, of course, cortical thickness and surface area. And they also have different way of how they developed and through the development, but also they have combined information for thickness and surface area is cortical volume, of course. But then if we can look surface and cortical area and thickness, we can obviously extract different information. For example, from thickness, we can just extract information about cortical layers, or layers of a laminar structure, or from surface area, we can extract information of columns in surface. And then we can use this information from two different features and different properties to understand, maybe to gain some understanding of neurodegeneration, different from just looking at volumes. And by reconstructing T1 scans, we can also extract many more information about how cortex is folded, some curvatures, and so on. So different information for which node that carry different information, different features that carry different information from the brain. And this is actually true, because if we look, so we looked at the cortical thickness and surface area and BVFTD and Alzheimer's disease. If you look, so color-coded are the allegiances with fronto-temporal and parietal lobes from different lobes. And you see that there are actually differences in the way how cortical thickness has changed from in BVFTD compared to surface area. But then there are also differences in between BVFTD and AD in terms of how thickness changes in one or the other one compared to healthy control. And similar is with the surface area for BVFTD and then in AD. So there is indeed different information, and there are differences when we look at these two composite measures. But I also told you that it's not only cortical thickness and surface area, we can look at folding and curvatures and so on. And in this study, that is a study with how these features change with age, and looking at for cognitive networks and how integrity of these networks changes over age from individual to individual, which is basically quantified by this term, which is called flexibility. So how whether the node similarity between nodes within this network and the rest of the network changes over age is quantified by this factor. And then there are obviously DMN constantly, the default mode network, the one that is involved in Alzheimer's disease. We constantly find that there is something within this network. In this case, there are obviously some nodes that show that they're constantly highly within that network, but there are some other nodes that kind of change their latency outside of the cortex. And then we are now coming to the Brace-funded study. So having all this in mind, we wanted to actually test this method that one of my collaborators developed for Alzheimer's disease, which is to look at something that is called biomarker from Alzheimer's disease. So you can just read maybe what is written in case that I will just talk what was the method about. So looking at hippocampus or temporal lobe regions or all these biomarkers of neurodegeneration, Alzheimer's disease, and then trying to find the rate of atrophy, but also classify patients based on biomarkers for Alzheimer's disease, which is different from the method I show you, like when we were classifying the patient, in terms of that in this case we can derive also temporal information. We can find stages where these patients are. If they are, so basically we can look at a very heterogeneous group of participants. They don't need to be very well characterized and have the same level of atrophy or CDR score. So we can just look at a huge amount of data and classify patients based on their level of atrophy, but also their stage. So we wanted to again apply this method in aging data, looking at different features extracted from T1 scans, from surface area, volume, cortical thickness, and so on, and see if we can classify our individuals in a way that we can also follow the stages or when certain feature will decay with age. But so looking at all 9, in this case, features that we can extract from T1 images. And this was somehow partially successful in terms of that there is no characteristic feature. We couldn't find actually stages, but what we could find looking at, in this case, network characteristics, not only in feature characteristics, looking at the interactions between DMN, these 3 networks that we also looked at earlier. There are 2 subtypes or 2 different clusters of these participants, and constantly subtype 1 is older than subtype 2. And I'm going to explain later why. So one is called immune response, metabolic immune response, and then the other one is called stress-resistant response. And this is the name we gave them based on, again, on their gene expression maps. We also looked like left and right in order to, if we can find differences or lateralization in this classification. And what we found is there is a lateralization, but only for visual networks here, because there is only one subtype in. There is only one subtype in right hemisphere, and there are 2 subtypes for all other for all other networks that we looked at, and also for left and right hemisphere. So this may look a bit more intuitive in a way. So if you are used to looking at the average of the, so on the right is, here is left and right and left again. And then Y is just looking at internetwork connectivity and level of how similar features within this network. So our subtypes are different on average even, because the subtype 1, this is young adult, and this is our reference group, and this is the level we expect to see in the connection of these networks. But then our subtypes are different. This subtype 1 looks more like a CDR1 or 2, like mild cognitive impairment, something like normative aging. But subtype 2 shows increased connectivity, which is whenever we have some increased connectivity, we refer to it that this is more like a compensatory response, and then brain try to compensate by engaging this network and then increasing activity or increasing similarity. And this is across all four 3 networks and subtypes. And so why do we call them a way how, like metabolic, immune, and stress response subtypes? Is that this, you use the same mapping genes to regions and then correlating gene expression with actually gene expression, level of gene expression. So gene that we looked at this case were longevity gene from database with in total 250 genes. Oh, sorry. I'm struggling with my there are a total of 250 longevity genes, but then only 9 in each of the subtypes are statistically significant, or there is a p-value with FDR corrected higher than 0.05. And all positive correlations are red, and the negative are blue. And most of them are very similar. So the gene that we find, function of these genes that we find is very similar, but those that are negative, that means that there is a level of gene expression in this region lower than expected. This is DNR repair, and these are related to metabolism and immune system. So that's why our division, so that's why our subtypes are named that way. And similarly, when we were looking at what are the node importance in gene expression maps. So these are significant node in both subtypes that map up to longevity genes. And in both cases, most of these regions and most of these nodes are from DMN network or central executive network frontal regions. And there is also lateralization. There are more important nodes in the left hemisphere than in the right one. So we don't know exact explanation currently how to interpret it. So, but obviously there is a DMN network, and regions involved in DMN network, they constantly light up, either as long that they are correlated, and there is a higher level of gene expression for longevity, or in these regions, or for Alzheimer's disease or longevity. So obviously this is the network that is most important for neurodegeneration and also supporting cognitive functions in healthy and Alzheimer's disease. And so I'm going to conclude now my talk with just these four points, which are that our aging subtypes do resemble AD patterns, and that normative aging looks like AD subtypes. But then that also structural features that we were selected and also networks are important in this pattern, not only size and not only volume or surface area or thickness. It is important what are interactions between and connections between features. And also that what we found is basically that genes do shape vulnerability because they do map onto region that we expect them to map. So what else we actually would like to look at is maybe track these aging subtypes longitudinally and maybe confirm our data, validate our data. Look at functional studies, fMRI, and myelin maps and PET biomarkers as well, so that we understand also if we can map using PET data, if we can correlate our finding with PET data, for example, and understanding, or tau PET or amyloid PET, to understand a network involvement depending on the modality. We also would like to include some environmental factors and see how our subtypes map up to cognitive training, sleep that we just talked about, and different, some of them that obviously improve outcome, cognitive outcome. And this is my acknowledgement for my collaborators at Aberdeen and Cambridge and my postdoctoral researchers here at Swansea. These are people like Tim and Timothy Ritzman and Alexandra Young, who applied for this grant funding when we ended up working together. Venia was a researcher. She at the project she was really vulnerable because she was sitting at the computer. She analyzed almost 1,000 images, and to analyze one image takes 20 hours. So that's really impressive work from her. And thank you for your attention.